All right. So hi, I'm Rosie. I was an intern for the SHI Lab at University of California, San Diego this summer, which is a bioengineering and bioimaging lab. I studied lung imaging for the diagnosis and study of bronchopulmonary dysplasia. So to start off, I thought it'd be good to kind of go over what BPD is as a condition. So in short, it's a breathing condition that's caused by poor lung development. It's very common in premature infants and affects about 10,000 babies in the US every year. It's usually diagnosed within the first few weeks of life by babies that are having trouble breathing. There is technically no cure, but following a treatment plan usually allows babies diagnosed with BPD to go through and survive and live long and happy lives. However, oftentimes it requires a pretty lengthy stay in the NICU. Even after going home, it requires a lot of treatment, medication, breathing support, and frequent checkups. It's usually diagnosed via an arterial blood gas test, which involves getting a blood sample and checking the oxygen and carbon dioxide levels within the blood, and that's just a way of testing lung function. They may also take a chest x-ray and check for scarring in the lung tissue. So our method of detecting BPD involves something called stimulated Raman scattering, which is a new microscopy technique that I'll explain a little bit more in the next slide. But in short, it detects the metabolism of BPD, so the metabolism of cells within the lungs, and it allows for increased subcellular red solution and sensitivity. So what is stimulated Raman scattering, otherwise known as SRS? So it's a pretty new microscopy technique, and it's based on something called the Raman scattering phenomena. And that is when incident light interacts with the sample and it undergoes inelastic scattering, which just means that the kinetic energy of the particles are not conserved. And that causes a shift in wavelength, which is known as the Raman shift. And that corresponds to the sample's molecular vibrations. And as you can see in this diagram, it actually uses two lasers called, one is called a stoke and the other is called a pump. The stoke laser goes through something called an electro-optic modulator. That's what EOM stands for in the diagram. And then it's really just a series of complex lenses and mirrors before it reaches the sample. The lasers create a local electric field around the sample, and it interacts with the molecular vibrations that I was talking about earlier within the sample. And when the frequency difference between the two lasers is equal to that of the molecular vibrations, Raman scattering is enhanced and the signal is stronger. And that's what we're able to see on our images. So looking at when we actually get our samples, we had to decide which sections of the sample we actually wanted to image. So BPD at its core is caused by damage within the bronchioles, and those are the major airways in the lungs. And that results in dysplasia within the alveoli, which are the little lung sacs, or the air sacs. So we identified our two main regions of interest, those being the bronchial and then the distal airway, which are just smaller little branches that go off of the bigger bronchioles. Both of those structures are responsible for gas exchange within the respiratory system, and that's the main area of concern with BPD. So this is a sample, a BPD sample, and I've just kind of shown you which areas we're looking at. So the upper area is the bronchial, and then the lower highlighted area is the distal airway. So before I show some of the images, I thought it'd be kind of helpful to go through how we interpret our images. So the first way that we look at these is through something called the optical redox ratio. And this has to do with two mitochondria metabolic coenzymes. We have NADH and FAD. So high NADH signals are associated with reduced cellular conditions, whereas FAD indicates oxidized conditions. The ratio provides information regarding cellular metabolism, energy production, and oxidative stress. So oxidative stress is a condition where there are too many free radicals or free radical molecules within the body and not enough antioxidants to fight them. and it leads to cell and tissue damage and has been shown to contribute to the progression of conditions like cancer. Its optical redox ratio is used, therefore, a lot in cancer research. And the most important thing to remember with this is that the amount of FAD is directly related to oxidative stress. And then kind of to back up the optical redox ratio, we look at lipid unsaturation. So lipids that are unsaturated cause kinks in the chains of fatty acids, and that increases fluidity of the membrane structure. That prevents the close packing of the lipid molecules and increases membrane permeability. The interesting thing is that unsaturated lipids also have antioxidant properties that fight and protect the cell from oxidative stress. So now looking at some of our images. So the top section shows the signal for each of these individual molecules. So flavin is a type of FAD, NADH, and those are the two that we look at the most. So looking at those two images, the flavin and the NADH, you can see that the NADH seems to be brighter, but it's kind of hard to tell just by looking at them side by side. So that's when we skip down and we look at the optical redox ratio. So in this image, we see some orange, some blue, different areas have kind of different signals, but we're looking at somewhere between 0.26 and 0.3 for that optical redox ratio. And then jumping over to lipid unsaturation or lipid saturation, we also see a big range, some kind of whitish areas, others that are more green orangey. So we're looking at something around 0.65 to 0.69. And then this is the bronchiole of the BPD sample. You can tell just by looking at the NADH and the flavin signals that those are much closer in intensity than the other two were. And then that's confirmed by the redox ratio, where we look and we see that the ratio is around 0.48 to 0.61. It varies in the different areas, but that's much higher than the 0.26 to 0.3 that we saw in the healthy patient. The lipid unsaturation is a little bit interesting because, again, we see a pretty big range, lots of different colors, but we're seeing 1.06 to 1.2, and that's also much higher than we saw in the healthy sample. So that indicates a more fluid membrane, so less control of what goes in and out of the cells, and also higher antioxidant production, which you would think would be a good thing, but it really does make sense because you're producing more antioxidants to fight off all of that oxidative stress that's being caused by the BPD. So this is the second region of interest, and we see pretty much the same exact pattern. Our numbers did not change much based on the region, or the ranges didn't change much based on the region, so that was interesting. But again, we see much lower values for lipid saturation and optical redox ratio here than we do in this, which is the second region of interest for the BPD sample. Again, the numbers are roughly the same, and that was something that we weren't necessarily expecting, so that was interesting to see. So like I said, we see much higher oxidative stress in the BPD samples, which was to be expected based on what we know about BPD. And the lipids unsaturation is also much higher in BPD samples. So now what we're doing is we're looking at using stimulated Raman scattering in combination with other types of imaging just to get as much information as possible for the study of BPD. So something that we are working on, or we're going to start working on. So we're looking at MALDI-MS, which is a type of mass spectroscopy, sorry. And using that in combination with the SRS technology, as you can see from this image, it provides additional information regarding the interaction of lipids, and then also the way that proteins are distributed around the bronchioles and the alveoli. And that gets pretty interesting because what we know about the population of cells going in and then coming out of the bronchiole, those cells are different. So we can see how the protein distribution varies with the different cells. So I wanted to thank HuBMAP and the NIH for granting me this opportunity. And then also my PI, Dr. Lingyan Shi, and my mentor, Anthony Fung, who's a PhD student. This opportunity really helped me to get a lot more lab experience. I hadn't had a ton in the past. I got to work on not only dry lab, but also wet lab skills. That was one of the huge benefits of this lab. It was so big. There were people working on different things. I got to learn a lot of new techniques. Coming into this internship, I wasn't really sure if I wanted to, I knew I wanted to go to medical school, but it was a matter of deciding between, an MD, PhD, seeing the research side of things. And also during this internship, I was actually accepted into medical school. So that took a huge weight off of my shoulders. And I was really able just to enjoy the experience for what it was. So that was really amazing. So yeah, if you guys have any questions, I'd be happy to answer them. And yeah, thanks for listening.